За мен е огромно удоволствие да ви представя професор Себастиан Хеслер, който е професор в Льовен, точно по невронауки. So, Sebastian, welcome on stage here. And yeah.
Thank you. Well, first, let me thank you for inviting me to this wonderful city. It's a really fantastic experience, and I've been visiting a bit yesterday, and I was really impressed. I really liked it. So thanks for having me. What I'm going to talk about today is actually a project where we've been built some hardware to interface with the brain. And let me first introduce the topic of why we would actually want to interface with the brain. In fact, this is a very promising area of research. It was the technology of the year last year, and that reflects, of course, that there's a lot of hopes that interfacing with the brain will actually result in many medical solutions. It's a very fascinating field. It's also getting a lot of attention because Elon Musk is active in this field, which is actually great because a lot of attention goes in there. But it's also really an enormous opportunity because it's actually really for patients, because it actually allows to develop new technologies that enable solutions that were not thinkable, let's say 20 years ago.
Yeah, how's it going?
And so in this example of a brain-machine interface, you see there's a device, a chip inside the brain. It's connected to a computer, and then actually just by thinking of speaking words, just by thinking of words, in fact, that person can, let me start this. Just by thinking of particular words, that person can actually convert brain signals to actually speech. And more recently, there's been even a more fancy version where actually, here in this patient.
It is good to see you.
So they added an avatar so she can communicate with this avatar to her partner.
Do not make me laugh.
So she's basically communicating with her partner just through her brain without actually using her mouth. And that is actually, there's a small but significant patient population that actually loses the ability to communicate. And of course you can imagine that this is really very, very difficult for these people. And actually with this technology, they can actually regain the ability to communicate with their partners and caretakers. And so this is, I think, really one of the most, let's say, extreme applications of brain-machine interfaces, but it's really showing you the potential of this technique. And so what's important to appreciate is that actually this patient has an electrode array. So this is a 2D array of electrodes placed on the top of the brain. It's just an illustration here, of course. And what's important to appreciate is that actually like each of these dots is one electrode. And the size of the dot reflects actually how important the information was that came from this particular dot to actually either detection or classification of thought about words. And what's important here is actually that you can see that different parts of the brain are actually important for different things. And so just sticking a needle in one part of the brain is actually not going to be enough. You actually need to capture the information from distributed systems to actually get the full information that the brain actually represents. And this is where a little bit that problem comes in, because you want to cover a large area, but you also want to have a lot of resolution, because the more resolution you have, actually the more signals you have, basically more information you can grab from the brain. So that means that you want to have high resolution. In this case, this is from a publication also relatively recently. And you can see here in this patient, there was this high resolution array with 2,000 electrodes was placed over the part of the brain involved in actually making movements. And you can see that each dot is one electrode, and you can see that actually how these signals really migrate, and actually having high resolution really gives you a lot more information. But then here's a big problem. If you go to 2,000 channels, you can see this is from an experiment where I used this array in a rat. You have 2,000 wires that you need to connect. And then in the clinical setting where this data was coming from, you can imagine that having all these wires, you can see it's like really, really bulky. So this is not something you easily carry around. So there's really some need for innovation there. And The approach that we are taking actually to address this is actually to solve what's called in the field the connector problem, is actually to do a trick, and to rather than using a passive electrode, we actually want to use an active electrode, which is simply an electrode that can sense the signals from the brain, but there's an additional component which is just a switch. And when you have this switch, you can actually go back and forth, and you can actually turn these signals. You can choose to record the signal or choose to not record the signal. Why is this useful? Because you can actually then do what's called time division multiplexing, which means that you can actually sample all the signals one after the other, while actually reducing the number of data lines. So you would actually first go and record, let's say, the first row of electrodes, then you record the next one, and so by actually switching between on and off state, you can actually assemble the signals in sequence one after each other, and so in that way we can actually do what's called time division multiplexing. Here's another, well, if you don't do this very, very fast, but just one after the other, just slowly, you can actually do channel selection multiplexing, but that's the most important thing, is the time division multiplexing, where you basically can sample the electrodes quasi-simultaneously, and by at the same time reducing the number of wires. We're not the first ones to ever think about this, so there's been examples in the field. This is a very interesting approach using basically CMOS-like technology. Problem with that, actually, it's very nearly impossible to fabricate because it's actually just thinned down silicon. Then there's graphene transistors, which have their own problems. Fabrication is a nightmare, and they're also not very stable. Organic transistors is even further away, and then this is another technology that's called flex on chip, where you basically take a passive array and you glue it to a CMOS pixel array, which is, I would consider this rather a hack, actually, because there's nothing active there. It's just trying to solve the problem of connecting the many lines to read out electronics. So collectively, all these methods actually suffer from high noise, low yield in the fabrication, and also limited area for CMOS-like structures. Of course, you cannot go to just very large arrays. So this is then where we thought, okay, let's innovate there. And what we've decided to do is actually use a technology that's extremely well established to produce displays. So these devices like these things. And so the backplane of these displays is just a plastic foil that has a bit of electronics inside. And so we use that technology, which is a very mature fabrication process, to make a first proof of principle 256 array. And we actually fabricated it, and we then, of course, want to show that it actually works in an animal. So we did some measurements in a mouse, where we actually collect spontaneous activity from the brain with our array. You can see that here in this example, we actually collect 16, so this is a 16:1 multiplexing ratio, which means we can actually record 16 channels with just one wire. And I hope you can appreciate that actually each one of these data lines, or each one of these electrodes, carries a slightly different signal, which shows to us, and we've of course done a lot of in vitro characterization as well, but for this is from the in vivo situation, showing that actually these signals are really separated, and there's no problem with leaking between channels. And so you can faithfully reconstruct information from all these 16 channels by switching through them just with one wire, reading it out. This is another example of the mouse, where in fact it's just spontaneous activity. You can see really how the information or how the activity in the brain is actually moving around. And this is entirely spontaneous activity, but we also did evoked potentials, where we actually evoke a reaction in the brain and then measure that reaction that the brain is actually giving. And so the way we do this is we actually shock the hindpaw of the animal, and if you do this, you shock, then there's a little electrical artifact from the shocking, and then you can actually see the signal emerging. Let me see. Signals then emerge and can be captured. I think the video is corrupted. Oh, doesn't matter. And so you can actually get these evoked potentials recorded, which is another demonstration of actually that this method is actually working at least in a mouse for now. And What's nice about this, of course, is that the brain does not, as I mentioned earlier, the brain does not encode information everywhere at the same time, but it's actually a distributed system where there's specialization in parts of the brain. And so if you just look at where do these signals come from that are evoked when you evoke the hind paw, in fact, it comes from a particular part of the brain, and that is known to be related to the hind limb movement, so the hind limb sensation. So here's actually, if you overlay this on the brain, this will be the part of the brain that we know that's previously shown to be involved in hind lip sensation. And so that's cool. We have a nice prototype, but actually one of the often underestimated challenges is actually that if you want to bring a product into the clinic, so if you're really serious about translation, you need to think very early about manufacturing as well. Why? If you want to make a medical device that eventually ends up in the human brain, you have very large regulatory requirements, which means you need to make sure that you can actually produce devices that are all the same, that are very reliable, that everything is actually working in the same way as you predicted. And that's why actually one of the main motivations to choose this technology platform was actually for that reason, because we are building on, as I mentioned, the display industry's backplane technology, which means that we can actually manufacture these devices already now in a foundry. And so here we actually take this array, that's the proof of concept, and we make actually a similar one, but now not in our own clean room, but actually now we're actually getting arrays from the foundry, and we just do post-processing to put the electrodes on top. And maybe, I don't know if some of you are interested in the technology, so these are indium gallium zinc oxide TFTs. They're basically a very mature process. And so when we get those from the foundry, we have a prototype of 256 channel. We now actually have also some with 3000 channels. So we're reading out 3000 channels with a multiplexing ratio of actually 1 to 32. And so this is really how it looks like. So you can actually get wafer scale production of these arrays, which is really a unique thing for us, because it's really changing the way, it's not like doing something in your university clean room, but really you can actually go to scaled manufacturing. And so here's the in vitro characterization. What's really cool about these, when you get things from the foundry, the uniformity is just enormous. So the transfer characteristics of these transistors is very uniform. They're all pretty much behaving the same way. And when you do things in the previous proof of concept, this was done, by a PhD student in the clean room by himself, and many areas don't work, and they have different properties and different things. And so here you really get like extremely uniform devices, which is exactly why we are choosing to go to the foundry. This is an exploration where we started to investigate the thickness of those devices, and in fact, they're all built on top of polyimide, which is just a polymer basically. And then, of course, the thickness of the polymer affects the flexibility. And if you're thinking about the human brain, it's a very squishy, soft material which has extremely low Young's modulus. So you actually want to make sure that it's very conformal. And of course, the thinner something is, the better it is conformal. And so here is just an illustration of different thicknesses where we actually can use, in humans it's shown that actually 30 micron is good enough to get reasonably good conformability. But of course, if you think about the mouse brain, which is much smaller, you will have much steeper bending radius. So in fact, but the 9 micron polyimide actually conforms even very well to the mouse brain. I should say that actually 9 micron is actually very thin because of course these are wafer scale process, so they're all sitting on a glass, and you need to remove the 9 micron foil from the glass without destroying it, and that actually is a bit of an issue. So at this point we're actually targeting more the 18 micron polyimide. So this is a nice one, but we've already started then to use this to actually collect signals also on the mouse, so to basically replicate the first proof of concept with the technology that we can now get actually from the foundry. So we actually have this is also validated. I should also mention that actually the technology itself, so the indium gallium zinc oxide transistors, TFTs, they're not cytotoxic, so we've done the first tests on that. According to medical device regulations, they're actually not causing any toxicity issues in the brain. And so with this, we will actually develop a technology that has large area coverage, high spatial resolution, high conformability, and due to the multiplexing we can actually reduce the number of wires, meaning that we can actually really address the problem of the connector, which is just for comparison, so the technology that Elon Musk is using or other startups are using, they're at most at 1,000 electrode channels, and this is pretty much where the limit is, more or less, because otherwise you end up having too bulky connectors and it just gets difficult even with nifty wireless and whatever compression methods. And so I think that this is actually a requirement for the front end of any neural interface. And then what's also important is that we have industrial scale manufacturing, which means that we can actually make those devices, many of them, in the same way, which is actually often forgotten and often overlooked, that this is actually critical for translation. And I haven't shown you this, but actually we've also, together with our colleagues in IMEC, developed an ASIC that would actually be able to do all the, steer the digital lines and actually collect the analog signal, amplify it, and so to actually have a corresponding ASIC that would actually drive these devices. And what's very nice is that actually we get to very, very low noise. There was a lot of skepticism at first because these indium gallium zinc oxide transistors, of course, compared to silicon, are have much less electron mobility. So you could argue, well, they might be noisy, but what's actually critical to appreciate here is that while these are kind of tiny arrays, but in the context of, let's say, nanoelectronics, these transistors are huge. So we can actually ignore the problem pretty much by just making transistors large enough so that actually terminal noise is the only thing that matters. So the overall system has extremely low noise. Actually, it's roughly an order of magnitude better than what was published before. And so then, I haven't talked about any of the biology we're doing. We're also doing quite a bit of biology, but this was mostly the work of Horacio, my PhD student, and Pauline. And this work was done actually in collaboration with IMEC by my team. And yeah, so thanks for your attention. If you have any questions, please don't hesitate to ask.